Thank you. I would like to thank the Academy of Sciences for organizing this truly amazing event and for allowing me to make a contribution. I have done a lot of the work that I'm going to show now together with my friends and colleagues here in France, like Olivier, Yves Freniac, and Alain Destex, many others. And so it's, of course, extremely embarrassing that I will give this talk in English. I'm extremely sorry, but I guess it's better for you and for me. So it will be about computer architectures, and I will start by saying a few words on computer architectures, but also on the technology that really drives computer architectures, because I think we are in a kind of an interesting situation where the technology is going to change very, very soon, and that will certainly have an effect on the way we build neural networks, for example, in hardware. So you probably all heard about the von Neumann architecture. Von Neumann was a theoretical physicist, and he's one of the pioneers of computing as well. And there is this architecture of which there are many deviations by now. It's not only the von Neumann architecture, but it's kind of the prototype for the computers that we use today. And the basic message is that computation and memory are separate. And in the memory, what we keep in the memory are the data and the instructions. We have two things there, the data and the instructions. And the content of the memory is addressable by the location. We know where the data and the instructions are. Then the instructions are executed sequentially unless the order is explicitly modified by your code. And the important thing is that they are physically separated. By physically, I really mean something that you measure in micrometer or something like that. So data and instructions, which are coded electrically by charges, have to be transported. And I put in this little picture there, which is the metal piece of an IBM microprocessor from the days when they still made microprocessors. And you see there is a lot of metal there that has to be charged all the time, which is part of the problem that the von Neumann architecture actually has. If we look to the top of such an architecture in terms of a chip design, what you see here is the scale of 1,000 nanometer, this little white bar there, which is a micrometer, and it corresponds roughly to the size of a bacterium. You see these colibacteria there. That's about the scale we are looking at here. And what you see up there is what's called a D flip-flop, which is basically a 1-bit memory, and it's realized in transistors that have a size of 180 nanometer, so 20% of a bacterium. And you see there are something like 10 transistors, there are 10 to 15 transistors that form this circuit. If you now get smaller and smaller, the same function can be crammed into 65 nanometer technology, which by now is also 5 to 6 years old, 28 nanometer, which used to be state of the art 2 years ago or so, and this little white dot there corresponds to 7 nanometer technology. It's all the same D flip-flop functionality, which is quite impressive. Now, what is more amazing and more frightening in a way is if you have a look on the vertical scale. Now, you don't look on top of the circuit, but you do a vertical cut and you analyze the system by electron microscopy, for example. What you see here is the transverse structure of a transistor, which consists of a gate, a silicon substrate, and then there is this layer in between, which is called silicon dioxide. This is supposed to isolate, electrically isolate, not to let electric charges through. And what you see, these little dots there, amazingly, those are atoms and molecules. You can count them, okay? I mean, the thickness of this layer, which is about a nanometer, and this is sort of really still an old-fashioned technology now. It's a 90 nanometer technology node. It's very, very small. It's a nanometer, and you probably believe two things. First of all, it's probably not a very good isolator because it's so thin, charges will move through. And the other thing is probably extremely hard to make this reliable. If you vary the thickness, for example, suddenly the isolation properties would change dramatically. And that is the reason why it's increasingly more difficult to build transistors, in particular to make them isolators, which are supposed to be most of the time. It's only if you switch from one state to the other that you are supposed to consume energy, which is unfortunately not the case because of these thin gates, which is one of the reasons. And what happened over the last sort of from 1975 to about 2000, 2010 was this amazing thing which is called Moore's law, which is not a law of nature. It's not like Newton's law, but it's really, it's an industry roadmap, and it doubled the number of transistors on a given surface, typically every 18 months to 2 years or so. But what we have seen since the middle of the last decade is a sort of a frightening development, which has to do with the effect that I have just shown, that the power of the transistor or the power of the circuits was limited. It flattened out. It didn't keep rising, which is good, of course. We're already consuming almost 100 watts on a very powerful microprocessor today. And of course, also the performance of a single chip sort of flattens out. There is still a rise of the number of transistors, which is good. So transistors can get smaller for the time being, but it will also end very soon. Now, around 2005, some kind of revolution happened in circuit technology. It is this black curve at the bottom where you see the black dots. Something starts rising very rapidly, and that's now the number of cores that we have on a chip. If a single core consumes too much energy, typically what we do is we build simple cores and we put many of them on the chip, and we exploit the fact that there are more and more transistors. Now, what we can expect until 2025, 2030 or so, that definitely the number of transistors will flatten out. It's the end of Moore's law as we know it, just because they will reach the atomic scale. You see they're already pretty close to the atomic scale. And what is also interesting, also the number of cores at some point will be limited because there is only such a big chip that you can build. And if you put more and more cores in it, they consume space. And at some point, you will also not increase the number of cores anymore. So people have seriously started to think about not only new devices, new transistors, but new architectures. And brain-inspired architectures are among them. And they do not necessarily depend on the smallness of the transistor, more of the idea for the circuit, which you may extract, for example, from biology, as we will see. Now, this is a view of a transistor, of a microprocessor. Again, it's an old-fashioned IBM processor. You see it looks like a city from the top with houses on streets, and that's basically what it is. It is a very modular structure. It is very engineering-friendly. You can design a system like that with many engineers working on different aspects like memory, compute units, routers, and stuff like that. It's relatively easy to map to programming models. We can all program these systems with the programming language that we learn at school or at the university, and it is theoretically sound. And traditional computing, as we have seen all the time, is very fast, very precise, very reliable, and it's numerical computation. It's, of course, very energy intensive. The power of a modern microprocessor reaches 100 watt. It has extremely high reliability requirement. We learned that we lose a neuron per second, or 100,000 neurons per day. That you cannot afford with transistors on a chip, definitely. So you need a very high reliability already during production. And it requires algorithms and software. If we look to a brain circuit, this is not a microscopic picture, but a reconstructed circuit that I got from Henry Markram in Lausanne, but it's on the same distance scale. And the first thing you see, it's not so clearly structured. There are no units. It's kind of uniform. So it is rather uniform. It has no obvious separation between memory and computing. It's definitely not programmed, and unfortunately, there is no established theory. There are many theories, but there is no single established theory like we have for the conventional computing. The brain is, of course, not a numerical machine. It's not for precise computing. It's for, as we have seen so impressively, for spatial temporal pattern detection and also for prediction making. It is very energy efficient. The brain is about 20% of a modern microprocessor. It's very fault tolerant. We heard about the neuron per second. And maybe most importantly, it can learn, and it does not rely on pre-compiled software. So what is the motivation for us to do this kind of work? Well, there are two motivations which we should distinguish. One is we want to understand what's going on in the brain, for example, in terms of learning and development. That is to support neuroscience in learning about the brain. That's one of the reasons why you want to build these brain-inspired computers. The other, of course, is very different to start with. It's future computing based on biological information processing. This is to achieve good performance in machine learning type tasks. And these things are not necessarily the same. They may be the same at some point. In both cases, however, you need something that I call a model system to test your ideas, to evaluate the ideas. And there are basically two modeling options. One is you write down a numerical model, okay, which you then put into code, and the model parameters are stored as binary numbers, like you know this from your computer science class. The other one is to do what we call a physical model, which is kind of partly, as we will see, analog hardware. The model parameters are physical quantities here. They are like voltage, current, and charge. And of course, what you can do is to build hybrid systems, as you will also see in my talk. The important issue, how much biology do we need? That's an interesting one. Do we want to build artificial brains? Certainly not. There's a lot of features, even of those which we know today, that are missing in all our circuits. But the observation is, and we have seen that before, that even dramatically incomplete aspects of biology, for example, just the fact that there are layers of neurons that are interconnected by weights with a certain strength, provide new approaches to information processing. You don't have to be biologically precise. You can extract simple things from biology. I have taken this from a very early paper here from the perceptron days, where people have implemented an XOR, which is a logical problem. It's a problem that cannot be solved by linear separation, just in the perceptron. And why am I showing this? It's interesting because biologists would say this has nothing to do with biology. There are no XORs, and also these neurons are not really biological neurons. Well, there are biological principles being used here. First of all, there are layers of neurons, but there is an interesting thing here. This thing only works because, you see, there is one of the connections which says -2, all right? That is inhibition. That really makes sure that if both inputs are on, the output is 0, okay? So the principle of inhibition, which is a very important one in biology, has entered into this very early phase of neural networks. Negative weights is a very important thing. So my claim is that if you put in more biology, we'll probably get more computational power. The question is, what does this curve look like the green curve or the red curve? We clearly don't know. Now, who gives us inspiration on where to stop, where to cut, what to put in from biology and not from biology? Well, the inspiration really comes from biology. And I've worked with biologists for a long time now, and it's absolutely mind-blowing what these people do, all right? This is modern neuroscience. It gives you access to multiple scales in space and in time. In space, the vertical axis, we go from synapses to the entire brain through units that are sort of coarser and coarser and less complex in their intricate structure. On the horizontal axis, we have time ranging from milliseconds, which is the synaptic time scale, to days and months and maybe even years, which is the developmental time scale. And it's interesting to see that these time scales cover 7 orders of magnitude, and I don't even go to the molecules here. That would be even more, and 11 orders of magnitude in time. And somewhere in this plane, and the colored circles and the colored objects show you the experimental methods that we have available to address those different scales. Somewhere in this, there is probably a sweet spot also for artificial computing, and we have to find this. I would, before I come to the actual implementations, like to show the importance of time. I will discuss the importance of the time domain. There is, what you see here is the simulation of brain circuits based on cellular models, where you really describe neural cells to some detail with ion channels and compartments and things like that. It's the memory requirement of the computational complexity. Computational complexity measured in petaflops, teraflops, and exaflops, and we now have typically 10 petaflop machines. That is the supercomputer that we have, and we can store hundreds of terabytes in a running program. And it means we now have, on the cellular level, access to smaller rodent brains. And what people say, for example, the Human Brain Project, now it's easy, we just go to exaflop, which will come to us sometimes in the next decade. We will have probably 100 petabytes of memory, or close to an exabyte of memory, and then we can simulate the human brain on the cellular level. That is probably true, but the question is it really useful? And I would say it's probably not too useful, because these simulations typically run a factor 1,000 slower than biology. So if you want to simulate a day of learning, for example, it will take you 1,000 days to get the result. If you want to simulate development of months, it will take you thousands of months. So that means all the time domain is not accessible in these kind of simulations, which is a very important thing. In particular, if you want plasticity learning and development, that would ask for another factor 10 to maybe 1,000 of computational complexity, which will not be available in conventional computers so soon. So one of the ways that we are looking into, and that many people in the world look into, is neuromorphic computing. And my definition is that here we implement some aspects, not everything, just some aspects of structure and function of biological circuits as analog or digital images on electronic substrates. But structure, I mean cell cores, the networks, the axons and the dendrites, the connections, which are the synapses. And the function, more importantly, is local processing in the cells, communication, and again, most importantly, learning. So what are implementations. Let me show you a few implementations, and before I do that, let me discuss a little bit the issue of digital versus analog, or maybe another solution. You know that all our computers that we use now, after a very early analog phase, are digital for very good reasons. Okay, we have discrete values of physical variables, computation by Boolean algebra, which is very nicely described and understood. One wire represents one bit of information, and the signal, most importantly, is restored after each gate. If you leave a logic gate, the signal is the same as when it entered it, so there is no degradation of the signal. In analog computing, you have continuous variable of physical, continuous values of physical variables, computation by component physics. Yeah, it's the properties of the physics of the components that determine your computation. One wire represents many bits of information, and very important, the signal is not restored after each stage. There is degradation, there is dispersion of the signal, which is very, very bad, because it means these systems are not scalable at all. And that is why analog computing had no success. However, there is nature, and nature, as we use it in our own brain, is something like, I would call that mixed signal. So there is local analog computing, there is local integration in brain cells of charges, but there is binary stereotypic communication by spikes. And by that, you achieve signal restoration. So you have energy-efficient local computing and signal restoration when you leave the computing stage. Now, there are several implementations. I will introduce 3 of them, which are ordered in degree of increasing exotics, I would say. This is the least exotic approach. It's the SpiNNaker project, which is also pursued at the Human Brain Project. This is called the many-core system. It's built by Steve Ferber, who is the inventor of the ARM processor. He contributed to the ARM processor architecture, and his mantra is, I want to connect many processors because they are so cheap. A processor basically costs nothing. What is expensive and what I have to work on is the communication between the processors. So he builds chips that have 18 ARM cores per chip. They run at a relatively moderate clock rate to save energy. They have some memory to keep the routing tables. But then, very importantly, each chip has 6 bidirectional links, which are like a little internet, packet-based information exchange, which can submit 6 million spikes per second per link, okay? And by that, you build something like what they call a real-time simulator. So it's an optimized simulator for spike-based neural computing. Then there is the approach by IBM, by the IBM Almaden group. It's what's called a custom digital design. So there are no longer microprocessor. There is no longer the computing model that we know, the programming model. The system is called True North. It's digital neural cells. So these are special circuits that can do only one single thing, that is to emulate a very simple neuron model, or very simple, very, very simple synapse model. There are 1 million hardwired, so-called leaky integrated fire neurons on a chip, 256 million crossbar connections, which they call synapses. Of course, these are extremely simple synapses. There's no on-chip plasticity, but it's very, very power efficient. The entire chip uses about 73 milliwatts, which is a very impressive system, and they presented some very nice results recently. Finally, the most exotic approach is the one where, as I said before, you combine local analog computing with binary information exchange. This is what we did in the BrainScaleS project together with Olivier and many others. It's a continuous time integrating cell membrane. So basically use a capacitor and a resistor and a battery to build circuits like that, which are easily described by this differential equation, which you all remember from school. It's a simple linear differential equation, which is easy to solve. It's just an exponential function. Of course, this is totally incomplete. In reality, of course, the neuron is totally nonlinear. It has this effect of a threshold, or of something where if you approach a certain voltage, a membrane voltage, suddenly the membrane voltage starts to explode and rise and produces this spike. But I'm showing this very simple linear equation just to convince you of the obvious fact that time in these systems is not coming from a clock that is driving the system from outside, but it's coming from the internal properties of the circuit. I mean, there's a capacitor and there's a resistor, and the product of both defines the time constant of the membrane. Now, of course, there are many other time constants in the system. In our case, neurons have something like 20 time constants, so you all have to look at them at the same time. But the message is that in electronics, capacitances are small and conductances are large. In biology is the other way. So if you just use electronics as it is and you play no tricks, you automatically get systems that are faster than biology, and there's nothing you can do about it. They just run faster because the physics imposes this on the system. These systems have actually been built partly in the Human Brain Project and before. This is a picture of the SpiNNaker chip. This is a picture of the BrainScaleS chip, which typically look like chips, what they look like. The difference is that on the left chip, you see a memory, and the neuron model and the physics of the neurons is stored in the numerical values that are stored in the memory. On the right side, on the physical model, you can actually point at neurons and synapses. They are physically there. There's no multiplexing. They are just directly installed. For example, the two white areas you saw on the right side are areas of about 50,000 synapses each. Now, the thing what you have to do is to build systems out of that. You have to build big boards. In the case of the BrainScaleS system, we use so-called wafer scale integration, and then you have to assemble these things into systems. So if you come now to Manchester in the UK or Heidelberg in Germany, you can visit these systems, which are probably the two biggest neuromorphic computing systems that exist in the world at the moment. On the left side, the Manchester system has 500,000 ARM cores. On the right side, the Heidelberg physical model system, 4 million neurons and 1 billion synapses. The Manchester system is a real-time system, so a day is a day or a second is a second. And the Heidelberg system is an accelerated system by choice of the physics parameters, so a day is 10 seconds, which is very interesting if you want to study learning and development. If you want to use those systems, they are operational and accessible to everybody who wants to work with us. It's interesting now to look at energy because that was one of the arguments we had. Now, if you have some kind of computational primitive, like you say, my reference operation is a synaptic transmission, then what you see that now in our own brain and your brain, the energy you use is about 10 femtojoule for a synaptic transmission. In a supercomputer simulation, like what Markram does in Lausanne or what people do with simpler models, it's typically 1 joule or a fraction of a joule. So there are like 14 or many, it's something like that, orders of magnitude between biology and the supercomputer simulations. The neuromorphic systems that are now operational, the SpiNNaker and the BrainScaleS system, they sit at 10 to the minus 8 and 10 to the minus 10 joule, which is already a big step forward. Of course, still far away from biology, but the important thing is that these systems are still configurable and can be read out. And that, of course, also requires a lot of energy, which you would not have if you would just use them as computing devices. As I said, more importantly, the time scales, okay? If you have a simulation, you have access to causality detection, synaptic plasticity, which typically act on the sub-millisecond to second level, which on supercomputer simulations is about a fraction of a second to 1,000 seconds. That's easily accessible. If you want to go to learning and development, it's out of the question for large systems. If you have a real-time systems like SpiNNaker, these time scales are easily accessible, certainly the learning scale. If you have an accelerated system like the BrainScaleS system, you can do developmental processing like rewiring of networks on the thousands of second scale, which is quite an interesting thing with interesting research perspectives. Let me show a use case or some use cases. The first thing you try to do, of course, is to do reverse engineered network architectures that you take from biology and let it solve a real-world classification problem. This, for example, is the olfactory system of insects, which many of you may know is based on chemical receptors that these insects have in their antennal lobe. They have receptor neurons, which are then processed by this network, which basically has two stages. There's a decorrelation stage, which is contrast enhancement by lateral inhibition, and there is an association stage where you actually combine certain chemical sensors to detect certain flower types, for example. And what you have to do by the learning is to train these connections between the decorrelation and the association stage. And we did that with this kind of system. And so what you get is these kind of images, which are a bit difficult to recognize here. But the little dots that you see, they are the spikes. This is the activity of the artificial network. And you see this kind of regular structure at the bottom. This is the input of the data to the system, and you see it's what we call rate-coded. So these neurons these are the receptor neurons. They fire like crazy because there is a lot of input. And that's very energy expensive, of course. I mean, if you have rate coding, because you have to pay for every spikes in terms of energy. And that's a very expensive thing. The same is true on the output, where you also use rate coding. The output neurons tell you if they did the classification successfully. But all the network in between almost never fires. It has a very low firing activity. And that's one of the reasons why spikes are so efficient, because with the sparse coding in the network connecting the input and the output layer, there's basically no activity. So that's very interesting. The two networks are before and after training. So typically what you get is a classification performance that is maybe as good as you do with software. This is a comparison to a Bayesian classifier with a 5-fold cross-validation. And this is the decision time in biological time scale now. On electronics, this has to be scaled back by a factor 10,000. And you see you reach some kind of reasonable classification on a time of 42 to 75 milliseconds. That's typically what you do with your own brain if you try to do this multivariate data classification. The performance is equivalent to classical machine learning, but of course the interesting thing here is that the energy efficiency is hugely improved. It's nanojoule, a fraction of a nanojoule rather than millijoule. The response time is microseconds rather than milliseconds, and the learning time is accelerated by a factor 10,000. So that's interesting, and this is one of the motivations why we are doing these things. There's an interesting observation, and that is if you look to the neurons, to the analog neurons, they're all the same circuits. Okay, they're identical circuits. There are hundreds of thousands of totally identical circuits. But if you measure them, they're all different. And that is, of course, a feature of electronics, of all electronics. In digital electronics and computers, you try to make the differences so small that you can easily distinguish between zeros and ones. Here we cannot do that. The variations are really, really large. These are the response curves of analog neurons, where we have the input rate versus the output rate. And they should all be straight lines, which they are, but they should all be the same, which they are not. There's a variation of 20% or so. And this is what I call spatial non-determinism. Okay? So neurons are all different, like they are on the brain. The question is, how do we handle it? Here, we handled it with so-called population coding, and I will tell you an alternative method in a minute when I'm coming towards the end of my talk. Let me, at the end, say a little bit on what I would call plasticity and the variability challenge of neural. That concerns now these analog neural networks in particular. This is a horrible picture, or a beautiful picture, I should say. I took from a biology publication. It's the complex protein organization at the postsynaptic terminal. This is a presynaptic terminal, a postsynaptic terminal. There is a gap in between where the neurotransmitters move. Of course, this is a biochemical process. In our circuits, we emulate that with electricity, but nevertheless, in nature, it's chemistry. And in particular, on the postsynaptic side, there is a very complex protein interaction going on, which certainly, almost certainly, contributes to information processing. So our current models of synapses are shockingly simple, I have to say. We can still do cool and interesting things, even with the plasticity, but it's clear that there's a lot that we haven't understood, but we should be prepared. We should be prepared to systematically explore what the role of this protein network is. Our current synapses, and this is a drawing, a chip design drawing of one of our synapses, is 10 by 10 micrometer. That's huge, okay? It's about 40 transistors. It's implemented in a rather old-fashioned technology node, 180 nanometer. Clearly, the size of these objects, and there are 1 billion in the wafer systems that I showed you, limits the integration density. If we can improve on the synapses, that would be great. On the other hand, we want to make them more complex because we want to be able to study the role of these protein networks. The weight that is available for learning, for example, is 4 bits, and there are various plasticity mechanisms, like short-term plasticity, depression and potentiation, and also spike timing dependent plasticity. Also, these synapses have variability. If you take many of them, I think there are something like there are 60 in this diagram, and you see these typical curves there. These are so-called STDP response curves. And on the right side is a single synapse. On the left side, you see 60 overlaid in one diagram. And you see, again, there is this variation of typically 25%. And you have to handle that, and you maybe have to exploit that for information processing. Now, there's an interesting way of improving on synapses, at least in terms of size. This is a slide I've taken from Julie Grolier. She works here in Paris at the CNRS Thales Lab, and she works on an incredible thing which is called Memristors. These are units that are non-volatile, non-linear, analog, and tunable nanoresistors, which would be ideal candidates for synapses if you can understand them and combine them with the classical CMOS technology. Why are they so great? Well, there is this little grid there, which I show. So the crossing points are typically 50 nanometer by 50 nanometer, rather than 10 micrometer by 10 micrometer. So there is an improvement in integration density of maybe something like a factor 100,000, which would be enormous if one could make that work. There are various technologies. You read a lot in the literature these days. There's phase change memory. There is really a configuration change of the chemical substances in the material, like the titanium oxide, for example. There are charge effects, and there are also purely electronic effects, like the orientation of spins. This is what Julie does in her work. And now these are candidates for higher integration densities, but of course, these things will also have variability, and measurements show that the variability will be even higher, of course, than the one from the CMOS circuits. So what can we do? This is one of my last slides here. There's a huge parameter space now that you have to adjust in these systems. That is the network topology, the neuron sizes and the parameters, and the synaptic strengths and the time constants. And currently what we do this with pre-computed values externally. Like, for example, also the True North group has just published a paper where they do learning, and they do it externally. They don't do it on the hardware, they do it externally, then load the system, see how it works, do some kind of backpropagation, and recalculate and evaluate again. That is a slow process. And so the next thing to do, very, very important, for me it's the only approach to ever access really large scale in these systems, is to go to what I call local on-chip learning. That means the learning replaces the calibration, which is in principle necessary for the electronic systems. So we do it like in biology, which is called homeostasis. We have plastic topologies and delays covering time constants from milliseconds to years, including the chemical processes, and cover automatically systematic variation of neuron synapse and learning complexity. And a systematic approach with local learning on chip, to me, is the only way to ever access the human level scale and beyond, in particular when you will use novel nano devices and learning. So I'm through now. I will just show you the first prototype where we now, in the Human Brain Project, explore these kinds of things, where we have neurons and synapses that are analog circuits, and we have what's called a plasticity processor that can emulate, for example, a lot of these processes that are carried out by the postsynaptic terminal, as I have shown on this figure before. My conclusions are that after 10 years of development, and that's what it took, we all started around 2005, IBM and also SpiNNaker in our own group, we have available hardware systems that have reached a very high degree of maturity and are ready for non-expert use. That's very important. Everybody can use them because there's also the necessary software to operate them. There is a high degree of configurability, which is very important with dedicated software tools, but of course it's not a replacement for general purpose machines. I think that is clear. It's implemented, accelerates learning, and that's the key for applications. There's no way you can just load a system and hope that it works. It has to go through a learning process, and the local learning is very, very important. And I think these systems are well suited in the future for the use of deep submicron, very deep submicron, in particular also non-CMOS devices. Thank you very much.
Let's have one question now. Let's wait. Yeah, Stéphane Mala.
You began by saying that the Moore law was going to flatten out, or there are prospects of flattening out, but all these architecture are 2D or layered 2D, and neurons are indeed 3D structures. What about prospects of 3D electronics?
Exactly. So first of all, neurons are 3D structures, and neurons have a very complex structure also. They have many compartments, and probably a lot of pattern recognition, in particular temporal pattern recognition, is carried out by the neurons themselves. And we are working on ideas of multi-compartment neurons that, for example, implement the hierarchical temporal memory model of Jeff Hawkins. But what you say is that the brain as a whole, of course, I think, is a 3D structure. And all our systems are 2D. So what we have to do is to map a 3D network to a 2D surface, which is not really possible. In particular, for larger network, it starts to be very, very hard. And so it's a very, very important development that we exploit what's called 3D integration of circuits. Solid-state companies are working on that. TSMC, for example, in Taiwan, starts to offer this also now for external users. And I think one of the very, very important next steps would be to make use of 3D integration.